some work in progress and some work that I've completed. I am, I guess a quick disclaimer, I'm actually taking my first geochemistry class this semester, and it's the first chemistry class I've taken in maybe 10 years. So there are some elements of chemistry that I'm going to discuss in here. If there's anything that you want to, jump in and give your input, like at any time, please go ahead. This is going to be pretty informal and, like I said, a work in progress. So the main motivation for my work is basically understanding where we might find habitable environments within our solar system outside of Earth. And the prime targets for these are these ocean worlds. And these are worlds that basically have ice shells that overlie what we think are global subsurface oceans. The two worlds that I'll be talking about in this talk today are Europa and Enceladus. Europa is one of Jupiter's moons, Enceladus is one of Saturn's moons. And the reason that I choose these two, it's kind of for two reasons. One is we don't really expect the kind of more exotic high pressure ices at these worlds. And we also kind of have some constraints on what the ocean composition might be. So that's part of the reason I'm focusing on these two. Another great reason is we're actually sending two missions to Europa to study what's going on there. So again, one of the reasons why we're looking at these worlds is because they're thought to host basically what the ingredients are that are thought to be necessary for life. Broadly, these are water, which acts as kind of a medium to support biochemical reactions, chemistry, which would be like the building blocks for life to grow and reproduce, and energy. And so by energy, I mean kind of food for organisms to basically live off of. So we think that potential habitats are going to exist where these ingredients will be found. So this is a figure from the 2016 Europa Lander report. And one of the things that you might notice is we kind of are highlighting regions that are potential habitats that are mostly coinciding with where there are water, first and foremost. And so the reason habitats within the ice shell are of interest in particular is because of their potential to be sampled by a lander. And they also represent targets for future ice penetrating radar investigations. These would basically be reflectors that we might see within the ice. So on the most fundamental level, the distribution of water and ice is governed by pressure, which in the context of ice shells will be governed by the thickness as well as the gravitational acceleration. The temperature, which will be governed by the geodynamics of the ice shell, so if it's convecting or not. The salinity of the ice, which we believe should be controlled by, in part, the ocean salinity, but also kind of how the ice is forming to make the ice shell. And I'll touch on that a little bit later. And then there's this question of composition. We think there's the potential that the ice shell composition may be representative of the subsurface ocean, but there's also this question of how do we expect that compositional signal to be fractionated? And I'll touch on that just a little bit, but for the most part, I'll kind of be assuming that the ocean composition is reflected in the ice shell. So if we look at our first thing, which is the influence of pressure, what we can see is that, again, there's not really high pressure ices, but that we do experience some depression of the freezing point based on this overburden pressure. So what I have here is kind of just like the phase diagram for ice that just shows where ice, vapor, and then water is stable. And so on Earth, we have, the gravitational acceleration that we have, which leads to like at maximum, maybe like 5 or 6 megapascals at like the thickest floating ice. And what I define as the thickest floating ice would be like the grounding line of an ice shelf, an ice shelf, which you could argue is not like totally floating. But that would be kind of an upper limit of what we would expect. Although the ice shells on Europa and Enceladus are considered, could be anywhere from kilometers thick to tens of kilometers thick, because the gravitational acceleration is so much less, we end up kind of within this region that's almost representative of what we would see on Earth. So one of the other things that can influence the presence of melt is the presence of impurities. And so what I have basically done is taken kind of some of the constituent ions associated with these different ocean compositions. So for Earth, we obviously know what the composition of the ocean probably is. For Europa and Enceladus, again, these are either modeled from kind of atmospheric constraints or kind of chondritic models. And in the case of Enceladus, we actually have a sample of what we call plume material, but basically material we think is from the ocean that was essentially spewed into space and sampled by Cassini. So there are some models for the ocean composition there. But broadly, we have sodium, chloride, sulfate. And basically what I did is I kind of just took each of these ions and examined the binary solutions for each of these. And so we have chloride solutions and sulfate solutions. And kind of the thing I want to highlight here is you might notice that the chloride solutions have a much lower eutectic temperature. And what eutectic temperature represents is the lowest temperature at which you'd expect melt. So that's kind of why it's relevant for this question. And then sulfates, much less freezing point depression, with the exception of ammonium sulfate, which I don't totally understand this behavior, but it's kind of a strange, it deviates from the general trend there. So the reality is the world is rarely binary. So we're gonna look at what happens when we put more complex solutions and we examine phase behavior there. So in the case of standard Earth seawater, we end up with finding like a eutectic temperature of maybe minus 36 C. And then kind of similar order to some of these other bodies that we're looking at. And notably, these are, excuse me, much lower temperature really than any of the binary solutions, maybe with the exception of this Enceladus. So I'll go back and show you magnesium chloride was kind of the closest to those at like minus 32. And these are a bit lower. Something else you might notice is these like insane contrasts, like all of a sudden in the phase behavior. This basically represents where you might have precipitation of salts as the solution is freezing. So going back to kind of this like fundamental assumption of like we expect some salts to be incorporated in the ice. What we think governs how much salt is incorporated in the ice from the ocean is kind of two things. One of which is the mechanism at which ice forms. So there's kind of like two families of ice that you can look at. One is congelation ice, and this is kind of the most intuitive ice that you guys might think of. It'd be like the ice cubes in your freezer, basically the directional freezing of water at an existing interface. And then frazzle ice is a little bit like less familiar. What this basically represents is ice that kind of crystallizes within a supercooled water column and then buoyantly accumulates beneath an interface. And so with congelation ice, you have this like one other kind of level of nuance, which basically says, depending on the interface microstructure, whether you have a very planar interface on a microstructure level again, as this is freezing versus like a cellular interface, will kind of govern how much salt is being incorporated and how it's being incorporated. So broadly, kind of what we found is that for congelation ice, we're going to be, oh, I completely forgot this part. One of the ways we actually represent how much salt is entrained in ice is something called the solute distribution coefficient, which is basically the salinity of the ice over the salinity of the ocean. What I was interested in is kind of like how the salt is incorporated at these very low growth velocities. where you might have this distinction being relevant. So on Earth, we have pretty high temperature gradients. And so this is kind of the dominant sort of microstructure that we see. So this would be like sea ice. So when I went to kind of understand how much salt is in ice, we basically came up with these kind of what we call equilibrium distribution coefficients of what is like kind of the limit as the growth velocity approaches zero. And so these were kind of the values that we found. For a planar interface, salt rejection is pretty efficient. So you get, I guess that would be 0.1%, 1%, 0.1%. Yeah, so basically the moral of the story is we are incorporating salts in the ice. How efficient that is just an artifact of how it freezes and the characteristics of the interface. In terms of composition, it's not a very well understood problem when you form ice, how representative that ice is compositionally of the subsurface ocean. And so basically what I ended up having to do was almost like a literature review of these different studies that were looking at these fractionation signals in ice. And so broadly what I found is for sea ice, there wasn't a very significant degree of fractionation. So basically the composition seemed to be representative of the subsurface ocean, with the exception of a couple minor things. We saw that like potassium overall tended to be depleted across a bunch of different samples. Magnesium tended to be enriched. And sulfate was the very strange one, where you tended to see regions of enrichment and depletion that you could basically correlate to where you would see flushing and refreezing. For marine ice, which is basically the frazzle ice that accumulates beneath ice shelves, we saw an enrichment of permanently, but again, very minor. In ice, where you would expect a very planar interface, this is something that we mostly get at in laboratory experiments, because again, we don't really see this for sea ice. We saw that there's an enrichment of ions that can be accommodated in the lattice. So that would be like chloride. The thermal structure of the ice shell, it will be, again, depending on whether or not the ice is convective or conductive. So some work that Clara Kalasova has done kind of demonstrated where we would expect melt using a eutectic temperature of 237 Kelvin, which is actually pretty close to what we'd expect for Earth's composition. And so what you can notice from this is in the case of like a conductive ice shell, basically your profile is just kind of stretched to what the total ice thickness is. And then if you look at the conductive lid, it's again kind of representative of like what you might see in just a conductive profile. So the focus of that I'm going to have is just looking at the conductive profile for now. So we can use all of these constraints basically to model the distribution of brine and ice. And we can do this by basically using pressure, temperature, and salinity as state variables, so long as we have an understanding for how the phase behavior of the solution evolves as it freezes. And so this is a problem that was addressed, almost 10 years before I was born. And the solution that these guys had was very elegant, basically posing this question the same way, using only temperature and salinity basically as state variables and representing the phase behavior of sea ice. So because we're interested in brine volume fraction, we need to account for the density contribution, not just the mass fractions associated with the different phases. So the density of ice as a function of temperature is something that is very well understood now. There's an organization called the International Association for the Properties of Water and Steam that has a relationship modeling the change in like thermophysical ice properties using this kind of Gibbs free energy formulation. So they take in all this data and then they basically fit it to a model that allows us to represent these ice properties. The two other things in this expression, F1 and F2, basically represent the phase behavior. And so this is basically the density and the salinity of the brine, along with these two, they're not really constants, but we're going to call them constants. They're a function of temperature, that basically define the ratio of the mass of salts in solid salts. So basically what that means is for hydrohalite, we have NaCl to H2O. This is just tracking NaCl. And the mass of salts in brine. And then the total mass of solid salts, so that would be both the NaCl and the 2H2O in the case of hydrohalite, over the mass of brine. And so this is stuff that's thought to be completely determined by phase behavior, so not necessarily dependent on whatever initial salinity the solution has. And we prescribe a bulk salinity of the ice, and this is something that you could kind of get at using the those equilibrium distribution coefficients I presented earlier. So in the context of sea ice, how this was done originally was basically taking a bunch of experimental data and then creating a phase diagram of it. And so this is a pretty famous phase diagram. You might often see it in the context of sea ice. And it's referred to as the Ringer-Nelson-Thompson pathway for the freezing of ocean water. There was some work that Giles Marion is the creator of the FreezeChem aqueous geochemistry software. And he basically showed that this data that was used to build this phase diagram represented a fractional crystallization pathway. So this is basically where things that precipitate weren't given time to kind of re-equilibrate with the brine and the rest of the system. And also slow kinetics. So again, it's an issue of time. Whereas this alternative pathway represented the equilibrium case. And so that's called the Gitterman pathway. And if you go back to why Azar 1958, why he used the data of Ringer, Nelson, and Thompson, it was because he couldn't find the data of Gitterman. So it wasn't that he thought this was correct, it was just what was available. So today we can actually use aqueous geochemistry software to model the phase behavior. And so this is a diagram that's basically representing this from a fairly recent paper that applies FreezeChem, that's one of these softwares, to the problem of sea ice, so ocean water freezing. And so we're going to go back and kind of rigorously examine this idea that F1 and F2 are determined entirely by phase behavior and are agnostic to the initial solution salinity. So if we go through and basically run FreezeChem for a bunch of different solution salinities, and then derive these two functions, we see that there really doesn't appear to be any sort of dependence on salinity. The behavior is represented here. What you do see is there's basically a point at which this function is valid, which is basically the freezing temperature. So that's, again, kind of what we would expect. This was like a little bit too nice. So we decided to look at more extreme salinities to see if this would break down. And it did. And so the question became, this is interesting, why would this happen? Or at what point would this approach then be valid? And why does it become invalid? So looking at this, it's kind of hard to tell, like, what is the thing that is breaking? Because you have brine density, brine salinity, and then these coefficients right here, function of temperature. So what I basically did is I went through and tried to figure out what was the culprit. So I plotted brine salinity and brine density, and these are both things that we can get out of FreezeChem. And saw that, again, it doesn't really change, and you get kind of the expected behavior where basically the freezing point is governing where this curve starts, which is as expected. So nothing really there. But here we actually found that this coefficient was the one that was changing, and it seemed to be sensitive to salinity above maybe 100 ppt. whereas this was not. And so a reminder of what this actually is defining is the mass of salt in solid salt and the mass of salts in brine. And so what I think is happening is that fundamentally from a mathematical standpoint, what you can see is that this term right here is lower when you have a higher starting salinity to the solution. So what can happen is that when you have a higher ionic strength at temperatures like above the freezing temperature, what this can do is affect the activity coefficients. And so this is basically the concentration adjusted for ionic strength. And so that increases the solubility. So what is probably happening is that we need higher concentrations to precipitate these salts. And so the question I kind of came to is maybe this really only applies where the salinity is such that concentration is maybe approximately equal to activity. I'm still not really sure, but this kind of makes sense to me. So the other question is, again, we're talking about the phase behavior in ice shells. So the pressure environment is obviously evolving. So does this really work when we're looking at things that are not atmospheric? So all of the phase behaviors are modeled using FreezeChem, atmospheric. So what I basically did is I took a look at, a simple binary solution first, basically examining, can I explain the change in at what temperatures brine is stable just by looking at the depression due to pressure melting. And so what this curve basically represents is the phase behavior corrected for pressure melting. Basically taking whatever delta T I expected from just the overburden pressure and applying it to this. And so what that ends up showing is that for the most part, that seems to explain the behavior. There doesn't seem to be like a significant change in where the salt actually precipitates. It just seems shifted by temperature. There's like a slight deviation with the very, very high pressure. Note that this is actually higher than the pressure we'd expect. So this is 100 megapascals, which if you recall, the Europa, I think, was about 50 megapascal at the highest pressure we expect beneath the upper limit ice shell thickness. So this is also a very dilute solution of NaCl. So I decided to look at a higher salinity case, and you end up seeing kind of the same behavior for the most part. But again, some weirdness at the very high pressure. There's like a little bit of a jump here. I don't really know what's causing that. That jump becomes even more pronounced when you go to a higher salinity. So it seems to be that for the most part, the phase behavior doesn't really change with pressure in these binary solutions. It just becomes shifted in temperature, except for something weird happening at pressures above what we would even expect. So I did this also for magnesium sulfate, just to make sure it wasn't the particular species that I selected. And there is a slight difference in terms of like, how much our eutectic temperature is deviating from just the pressure melting curve. So this would be another way of looking at it, is how much can we express the change in eutectic temperature with pressure melting. So here we have the cases that we just looked at, basically represented in this way. And then for magnesium sulfate, the same. Noting that there's like a little bit of a change. Right here, it's a little bit more obvious. It's a little bit above the curve. But again, close enough. So these, again, are simple binary solutions. So then the question is, okay, what happens when you have a more complex solution? So I basically did exactly the same thing, basically saying, okay, let's just correct the pressure influenced by the pressure melting curve. And it like looks pretty good for the most part, again, except for the 1000 bar. But if you look at how the eutectic temperature is changing for these different pressure environments, for Earth, it doesn't seem to be as sensitive. And it is it's one of these things where I'm not sure if this is a real physical behavior or if this is just an artifact of the model convergence issues, because when you have a more complex solution, you're more likely to run into these issues. So this is kind of curious and conflicts with kind of the more simple behavior for the binary solutions. So this is something I need to look into a little bit more. So basically, to apply this method and feel comfortable about it, the solution or the assumptions that I made moving forward was, let's not prescribe a bulk isosalinity greater than 100 ppt, which is a pretty reasonable assumption for most of the bulk ice shell, just given that such a small fraction of salt is actually incorporated into ice while the ocean freezes. And then also just kind of ignoring this issue of pressure just for now. The phase behavior, we just derive it from a single freeze chem run, because as I showed before, like you could use a solution of 1 ppt, allow it to freeze, and basically derive all of the phase behavior from that. And then because you don't really have, your brine salinity is only defined up until the point that you really start freezing, like the equilibrium brine salinity, we basically say, okay, at 0 ppt, you would, that corresponds to like a 0 Celsius melting temperature. And that lets us get basically these parameters over the full temperature range that we're interested in. So I basically tested this for kind of simple binary systems. And the results are kind of shown here. The two things that I'm showing is how the brine volume fraction is changing with temperature as a function of the prescribed bulk ice salinity, and also like how it differs for different solution compositions. So the big takeaways here are sulfates tend to precipitate early in freezing. We kind of already saw that before with just kind of the much higher eutectic temperatures for sulfates. The other thing that's interesting is that the larger the bulk ice salinity is, the higher the brine volume fraction at the eutectic is. So that's like a pretty significant contrast between here's brine and here's where nothing theoretically should be stable in terms of brine. So again, ammonium sulfate is just bizarre. So if we take this approach and then apply it to Earth, which is kind of like where we originally got this approach from, we end up seeing that we, using FreezeChem, represent the current model that's currently in use fairly well. Something to note is that this model that we use for sea ice today, it actually uses the phase behavior from the Ringer-Nelson-Thompson pathway that I showed before. The thing that I've highlighted here at -5C and 5 ppt is what's called the golden rule of fives, which is basically at this salinity and at this temperature, we reach a porosity of 5%, which is thought to be the permeable impermeable transition for ice. So this was kind of for brine and ice, which is was a good like sanity check for me. This is kind of again representing what we think. The discontinuities that you see here are where you have like more major salts precipitating, like a higher amount. And notably, like the equilibrium freezing that we represent in FreezeChem, it matches the old stuff pretty well, but deviates below this point. So if this were to continue to temperatures below what we would even expect sea ice to be at, you would see more of a deviation. So the other thing I tracked was basically how the composition was evolving as the brine froze. And so this makes sense. You see that there's really no change up until the point where you start precipitating sulfate. And so that's where you get a more and more fluoride enriched brine. And then again, you have sodium, hydrohalite precipitating. So that is a source of chloride, a sink of chloride. And so you see the sulfate to chloride ratio going up. and then back down again. And so that kind of shows how the brine chemistry is a little bit controlled by how these salts precipitate. So we applied this to Europa because we felt comfortable with our results from earth ice. And I just described 1 ppt as kind of an arbitrary bulk ice salinity. And interestingly, you actually see kind of this same behavior with the chemistry, that as the ice is freezing and the brine is getting more concentrated, you're ending up with a more and more chloride enriched and sulfate depleted, basically brine. And we can also apply this to Enceladus. The models that I'm using are from Mark Fox-Powell and Cousins' paper. It's a really, really great paper. I recommend it, where they're basically looking at, to some degree, the influence of pH on what ends up precipitating, so how that influences the behavior. And we don't have sulfate in this model, so I instead kind of use like a form of alkalinity, I guess you could call it, to track basically what's happening with the chloride again. This is kind of the second most dominant component in the brine, which is kind of why I picked it. And again, you see this kind of progressive chloride enrichment. Yeah, and so for pH 11, you'll notice that the eutectic, I might have pointed this out, is a lot lower temperature. This is in part due to the role that ammonia plays in the solution. So we'll take a look at that. So this is basically a diagram that represents how like ammonia speciates depending on pH. And if you look at the pH 9 case, you see that it's a little bit more ammonium than ammonia. And if you look at the pH 11, it's almost entirely ammonia. And so that is likely what's playing a role in the change in eutectic temperature between these two. So the presence of ammonia in ice is actually kind of important because of how low that eutectic temperature is. It's like minus 100 C. So this is kind of an interesting result because it's telling us that like even though there's ammonia there, we're not reaching that ammonia eutectic temperature. And I'm not really sure if that's an issue with the model convergence because it can stop when it reaches the eutectic or when it really just can't keep going. And so that's kind of an open question that I need to talk to Mark Fox-Powell about. So the major takeaways of kind of these results is that the temperatures where salts precipitate coincide with discontinuities in the brine volume fraction slope of temperature. The eutectic temperature of these multi-component solutions is lower than those of the individual binary solutions for the most part. Brines tend to become enriched in chlorides as they freeze. And this question about like, if you have ammonia in solution, do you expect that brine to be stable to the eutectic temperature of the binary aqueous ammonia system? And so one of the reasons this enrichment of chlorides in brine as these brine pockets freeze could be an issue is basically something called chaotrophicity. And so there's an idea that when you have enhanced chlorinity of solutions, you're actually going to decrease the stability of proteins and make it harder for these environments to be habitable. This is like a little bit simplistic of a perspective. So again, Mark Fox-Powell has this really wonderful paper that he published in 2016 that kind of looks at the different things that matter for the habitability of a brine system. And so water activity is one of them. And what this basically means is water that life can use, water that's not trapped by other things. And in high salinity environments, this limit is about like 0.611, and the absolute limit from another reference that Mark Fox-Powell cited in his paper is 0.605. So like that's what this red line and these figures are representing, is this threshold of water activity that would make an environment habitable or not. And so these symbols that are not filled in, as opposed to the other ones that have colors, represent the environments that are basically not habitable, so we think. So the other thing is this chaotropicity, which he represents as chaotropic activity. And so this is, again, something that causes like the breakdown of these systems in life that we think are kind of important for their ability to survive, basically. And so most of the solutions that were analyzed are actually cosmotropic, which means basically stabilizing for these macromolecules, with the exception of these two. So that was kind of an interesting result. And ionic strength is another thing that can influence the habitability of brines through kind of perturbing native structure and function is the way that he put it in his paper. And one of the other interesting things he highlighted is that you can actually look at the ratio of divalent to monovalent ions to look at kind of like how much this makes the habitat challenging. And this just really does tie back to ionic strength. So obviously it's like a little bit more complicated than just tracking the chlorinity of the solution, but it was a start. So the one other thing that's kind of interesting in the context of habitability that we can look at with these brine volume fractions is basically recharging them. And I mean kind of two things by that. The first is this idea of delivery of surface material to the subsurface. And we think that there's a bunch of oxidants being generated at the surface of Europa, which basically serve as a source of food, for lack of a better term, that if those materials can get down into the ice shell interior in the ocean, then organisms can use that along with reductants from the ocean. And so Mark Hesse has done a really interesting study where he's looking at the ability of porosity waves to deliver these oxidants to the ocean. But there's this assumption that these small melt fractions that we expect need to be able to percolate through the ice shelf. And so there's a real question of kind of how permeable is the ice shell and how does that translate or how does that relate to the brine volume fraction. And so this plot here is from a study of multiple sea ice samples that basically represents how the permeability changes as a function of basically brine volume fraction. And so there's, again, this idea of a critical porosity of about 5%. And you'll notice that like there at least appears to be somewhat of like a drop off, but clearly some degree of permeability even below this critical porosity. So I think this is a really important question that needs to be pursued. So the other element where permeability is kind of, or brine volume fraction and permeability is important is this idea of nutrient replenishment from the subsurface ocean. And so if you look at congelation ice, so that's kind of represented by the skeletal layer here. And again, that's that directional freezing of ice. You mostly have exchange controlled by convection and brine drainage. And this is traditional kind of what we think of as sea ice. Where you instead have these platelet ice layers. So this is again that frazzle that nucleates in the water column and then buoyantly accumulates. You end up with a way more porous ice. And so what's interesting about this very porous ice is you can see that it actually gets recharged on the scale of tidal cycles. And so this would be really interesting if this was something that was operating on Europa. So it would suggest that you could have replenishment on the order of the tidal cycle of Europa. And so basically what I ended up doing is imposing a conductive profile to represent Europa's ice shell. I set the subsurface temperature to the mean equatorial temperature. The basal temperature was defined by what you'd expect the freezing temperature of that subsurface ocean to be. And recall it's 12.1 ppt. This is like a third less than Earth's ocean, which is why the freezing point depression is like half a degree. The model for the steady state thermal profile I used basically assumes that a temperature dependent thermal conductivity of 1/T, and I neglected pressure melting. And so if you look at kind of the distribution of brine that results in this kind of admittedly simplistic case, you end up seeing that you really only have thermodynamically stable brine and maybe the bottom 13% of the ice shell, basically where the temperature is greater than this 240 Kelvin eutectic. Something interesting that I observed was that at no point does this like low salinity and number case. So I looked at 3 bulk salinities. The lowest one, at no point does it exceed this 5% critical porosity. I think this is something that's kind of interesting and maybe can be looked at in the context of constraining what the stable bulk salinity of the ice should be from a thermodynamic perspective. So brine volume fraction can control a number of other bulk ice properties, which can influence processes that are important to the distribution and stability of habitats in the shell. So these include bulk density, thermal conductivity, heat capacity, viscosity, dielectric properties, and structural properties. The code that I currently have and the program that I'm developing includes these 3 effects. So basically, what we have is a tool to give you profiles of bulk density, thermal conductivity, and heat capacity for ice of any composition over a range of temperatures. So how can future missions constrain the distribution of these potential habitats? I obviously work with the Reason instrument on Europa Clipper with Don Blankenship and others. So something that's always on my mind is what does this mean for detection? So basically these brine volumes and contrasts in brine volumes represent potential reflectors that will help us identify where these habitats might be. The magnetometer could constrain potentially the ocean salinity and thickness of the shell, which might be able to allow us to place internal reflectors in context of total ice shell thickness, which is important for us to understand kind of the coupling to temperature in the absence of an ice ocean interface reflector. So if for whatever reason we couldn't actually see to the ice ocean interface. And spectrometers could locate regions where salts may be concentrated at the surface, which could suggest the presence of like relict brine systems where there may have been habitats. So conclusions, these brine pockets represent potential habitats within ice shells of ocean worlds. The bulk salinity composition and thermal profile will govern basically the distribution and size of these pockets, amount of these pockets. The chloride enrichment of brines could potentially challenge the habitability of brine pockets in the ice shell interior, but this is obviously something that I need to kind of dig a little deeper. The permeability of in-ice shell brine systems are potentially critical to supporting these habitats, as well as delivering oxidants to the ice shell interior. And observations from Clipper could constrain the distribution of these habitats and other parameters that are relevant to habitability. So these would be like things that are governing convection. So yeah, thanks for listening.
Awesome. Thanks, Natalie. It looks like we've got a pretty big polar planetary group here. So hopefully we've got some good questions for Natalie. You feel free to just unmute yourself and ask.
Hey, Natalie, this is Duncan.
Hey.
So you've described the habitability from life being basically a passive participant in this. For these processes on the Earth, does the existence of life microorganisms mitigate these processes in any way?
Absolutely, yes, that is such a great point, Duncan. It's not something that, it's one of the things that I kind of need to look into. And it's definitely been shown that life has a way of kind of like altering its environment to basically allow it to keep going. I think a lot of the work that Mark Fox Powell did in terms of looking at these brine systems and what they mean for habitats is kind of, yeah, like more of the perspective I approach the thermodynamic one. But you're absolutely right. Life can alter the environment to make it more favorable to its survival. We know it happens since the ice, but it's not something I discussed here. You're totally right.
Thanks.
I have a question, Natalie. It's Kirk.
Oh.
Just when you're start precipitating these salts at the bottom of the ice shell and things start falling out of the brine, do you have any idea on if they would be detectable with reason, or are they too close to that, or too close to that attenuation would take them out, or they're too weak to actually be seen?
I think the latter. I think the latter. I think it's a really interesting question and something that I'm going to be looking into, of like depending on the composition of that underlying brine, it may influence, you know, like how much attenuation we might expect up to that interface. And so again, it would be simplistic looking at binary solutions, but yeah, like I think it's weird because so chloride in ice enhances signal attenuation with radar, but you also have that much lower eutectic temperature, which suggests that it would actually be, you know, closer to the ice surface, whereas sulfate, which, you know, doesn't really significantly contribute to ice shell attenuation in the context of electrical conductivity. So it would be less attenuation, but there's like barely any freezing point depression. So it would be so close to the ice water interface that, yeah, so not as favorable in that regard. So sorry, I went off on the tangent, but yeah, I agree with your later statement. I think it would be challenging to see.
Hey, Natalie, this is Don.
Hey, Don.
You were moving fast, so I was having
sorry.
I was having a heck of a time because I was trying to keep the Devon. For everybody that doesn't know, we've got Anya. Anya found a hypersaline lake under the Devon ice cap that we're challenged to sample some way, either by drilling into it or tracking the drainage from it to the coast. So I was trying to think of what it meant, because Anya's got a salinity for that thing. Where are we with regard to freeze chemming various thickness of that lake body?
Where are we with governing?
It strikes me that everything you have out there will tell us whether we have precipitates in that lake body, given a starting salinity and given an assumption about the distribution of the distribution of the components.
So yeah,
is it pretty much there?
So I think, and Anya, correct me if I'm wrong, you looked at like sodium chloride as kind of the brine composition, and then sorry, my dog is whining.
That's Alfie. I recognize him anywhere.
No, it's blue.
Oh, I guess not, huh?
So in the case of a binary solution, you basically are starting to precipitate these salts at the eutectic temperature. So in that like simplified system, you don't really have that. But again, because the world is fairly binary, there's probably other components in there too that would cause the precipitation of other salts. So I don't really know, I guess is the answer. I don't know if how useful that will be applied to that problem.
Yeah, well, remember, we're within 24 months of nominally getting some kind of sample of it, so we better have a hypothesis.
We can definitely work it. And yeah, sorry, I did go really fast. Part of the reason I wanted to give this talk was to establish a deadline for myself to look into some of these issues. So a lot of this was the journey I went on exploring these problems, and I'm really grateful that you guys hung in there with me and let me air out my insecurities for some of this stuff.
I have one other question.
Yep.
Yeah, still have time. As someone unfamiliar with ice freezing processes, you talked a lot about permeability. Is there a difference in how ice freezes on between horizontal and vertical permeability?
That is such a good question, Kirk. Yes, there is. And part of this has to do with the microstructure. Sorry, I'm still in. When you have that directional freezing, you end up with these kind of like elongated crystals. And so you have an enhanced permeability basically like along those like crystal plates, whereas the horizontal permeability is reduced. Off the top of my head, I don't remember the numbers, but there's definitely anisotropy there. Whether that anisotropy would be the same for like these like frazzle ice, I don't think it would be. I think a lot of it is related to kind of that inherent microstructure that's originally created and then kind of like propagates.
Cool, thanks.
Any last questions? Okay, well, thanks, Natalie. You definitely covered a lot. I learned a lot.
Sorry.
So thanks for coming, guys. See you next week.
Thanks all.